All right, let's do this. Today, we're going to crack the code on a real GCSE biology question all about plant hormones. We're going to use the official mark scheme to figure out exactly how to lock in every single mark.
I mean, it's the big question, isn't it? What's actually going on in an examiner's head? Well, today, you're getting an insider's look. We're going to pull this question apart, piece by piece, so you can see exactly what gets you the points.
Okay, let's get right into it. This is the one we're tackling today. Question 03 from an AQA higher tier paper. And it's all about plant hormones. You can see the setup right here. We've got some seedlings, a lamp, even a radiator. It's a pretty classic investigation.
Right, first things first, we're diving into the beginning of the question. And trust me, these are the kinds of marks you want to grab quickly, get them in the bag, and feel confident.
So, the question is straight up. It tells us gibberellins are used for germination, but then asks, what are two other uses? You've got 5 options here. Tissue culture, forming roots, increasing fruit size, killing weeds, or promoting flower production. All you got to do is pick 2. And boom! Here are the answers, straight from the mark scheme itself. 1 tick for to increase fruit size, another for to promote flower production. That's it. Nice and simple. 2 marks secured. Let's keep the momentum going. Okay, so the next part of the question kind of switches gears. It's less about just knowing a fact and more about thinking like a scientist. It's asking us to look at that experimental setup and figure out how we could make it better. It's time to put on your lab coat for this one.
So let's break this down. The question asks us to describe 2 improvements. The key things to notice are the command word describe and the number 2. That means 2 distinct points for 2 marks. You've got to think, what else could be affecting how these seedlings grow besides the light? What are the control variables?
Now check this out, because this is a huge exam tip. The mark scheme gives you a whole list of possible answers. It says any 2 from, you could talk about keeping the temperature the same, using the same number of seedlings, making sure they start at the same height. You could even say use more seedlings for a more reliable result. This is great news. It means there are lots of different ways to get those 2 marks.
Okay, let's think about another little practical problem. The seedlings aren't shooting straight up, right? They're bending and curving towards the light. So how on earth are you supposed to measure their length accurately with a normal stiff ruler? It's just not going to work. What would you do?
And that is exactly what the next question is about. For 1 single mark, you need to suggest a way to measure the length of this little curved seedling right here. And the answer is actually pretty genius in its simplicity. The mark scheme loves this one. Just take a piece of thread or string, lay it carefully along the curve of the seedling, and then measure the length of the string with a ruler. Perfect. Of course, you could also say something like, use a flexible ruler. It's a simple solution, but you gotta know the trick.
All right, we've arrived at the big one, the 3 marker. This is where you can't just throw out a single keyword. You have to build a logical, step-by-step explanation. This is where showing you really understand the science is going to pay off big time.
So, first things first, let's get our bearings. The question focuses on the difference between side P and side Q. Side P is right up against the lamp, getting all the light. And side Q, it's on the other side, hanging out in the shade. So, to put it simply, more light on P, less light on Q. Got it? So how do we build that perfect 3-mark answer? It's like a 3-step combo. 1, you state the obvious. Side P gets more light. Easy mark. 2, you bring in the science, the mechanism. This causes an unequal distribution of the hormone auxin. And 3, you explain the result. Auxin builds up on the shaded side, side Q, causing more cell elongation there. See how that flows? 1 point leads to the next, which leads to the final result. That's a perfect 3-mark answer.
Okay, we are on the home stretch now. The very last question moves away from all this auxin and phototropism stuff and tests what you know about a totally different, but equally important, plant hormone.
This is such a classic real-world question. I'm sure you've heard it before. Don't keep bananas in the fruit bowl if you want your other fruit to last. Well, for 1 mark, you have to explain the science behind that. Why does it happen? And the answer is just 1 word. That's it. Ethene. The mark scheme couldn't be clearer. It's looking for the keyword ethene. You just need to state that ethene is released from the bananas. That single piece of knowledge gets you the mark. Done.
So there we go. We've completely broken down the question, and we've seen exactly what the answers are. But what's the big picture here? What are the key strategies we can take away from this and use on any exam question they throw at us?
Here it is, your new toolkit for top marks. First, always spot those command words, like explain or describe. Second, know your keywords cold. Things like auxin and ethene are non-negotiable.
Third, when you see a phrase like any to from, just relax because you've got options. Fourth, for those bigger multi-mark questions, build your points logically, one after the other. And finally, the biggest tip of all, use the mark scheme in your revision. It's your ultimate guide to what an examiner wants to see.
And that really brings us to the end. When you start to think like an examiner, everything changes. You're not just learning facts anymore, you're learning the strategy of the exam itself. So the real question is, now that you've got this inside look, how are you going to use it to supercharge your own revision?